Okay. Hello, everybody. We're so glad to have you here today. And we're so pleased to have Dr. Aaron Miller joining us today to talk to us about the research at his laboratory and really about how the science is growing like crazy and trying to figure out the role of the microbiome in human health. And a lot of this is based on some new techniques that have not been in play for all that long, but it allows us to look at the whole community. So one of the things that I want to do at first is read this little biography of him so that you understand the context. Dr. Aaron Miller is a functional microbial ecologist and head of the Stones Translational Research Laboratory at the Cleveland Clinic. The focus of his research is primarily on how the microbiome promotes or inhibits disease processes, particularly for urinary stone disease. His work involves the use of multi-omics experiments in the laboratory and in the clinic to establish connections among the host, microbe, and metabolism. So it's a wonderful putting all the pieces together. So the goal of this research is to develop a novel diagnostic and therapeutic options to better identify individuals who are at risk for recurrent urinary stone disease and treat them through targeted bacteriotherapies designed to restore vital microbial functions that prevent the onset of the disease. So, but he and I have been talking for a long, long time, and I want to let you know that I can't exactly remember what was the first time that I talked to him, but it might have been when I read his study about wood rats. And so you can talk a little bit about that, but it was fabulous because what he was doing is going into the desert and looking at these rats that the only thing they could eat was really high oxalate. So he thought they might have a different looking microbiome. And I just, the moment I saw that, I knew that I would love his work. And so that continues to this day, Dr. Miller, and we're so pleased to have you. And what I want to do is kind of give you the throne here. And let's see, how do I do that? I've forgotten.
Um, let me see if I can share my screen, I guess.
Yeah, you did. Yeah, you did your share screen. That's it.
Well, so it says, so I think you have to, I don't know if you have to make me.
Oh, I have to make you a co-host. Yeah.
Yeah.
All right. Participants. I've got to find you on my list here.
Okay.
Now you can take the screen and thank you so much for being here.
Yeah, thank you, Susan, and thank you everybody for joining me today. So I am going to be talking a lot about these desert wood rats today. So really, what I wanted to do is kind of go give an overview of our research into gut microbiota dependent systemic effects of oxalate. And on the bottom left here is our desert wood rat, the Neotoma albigula. And then on the right, this is from one of our studies, and what you see illuminated there is calcium oxalate in an atherosclerotic plaque. And I'll talk a lot about that in a little bit. So just a brief outline of the talk. So I'm going to spend about 45 minutes actually talking about research and specifically, some of the interactions between the gut microbiota and dietary oxalate. And then I'm going to talk a little bit more about how this relates to systemic effects of oxalate and some of the important features of the gut microbiota that maintains oxalate homeostasis. Then after that, I want to spend about 15 minutes to open up the floor for questions for everybody. So before we get started, I want to make a plug for it's an NIH-funded clinical study that we currently have ongoing to longitudinally track the gut and the urinary tract microbiome over a 5-year period. This study is really focused on looking at the antibiotic effects on oxalate homeostasis and kidney stone formation, as well as other oxalate-related symptoms. And so if you are interested in joining, you can email lcmustudy@ccf.org, or you can text or call these two phone numbers that I have listed here. I have posted this flyer on the TLO Facebook group previously, and we can kind of revisit that again if needed. So I see this talk as sort of starting a two-way interaction between myself and people that are interested in oxalate metabolism. And so really the goal of my research is designed to improve patient outcomes. But this really requires patient input and participation in clinical studies and providing samples for this. So, I mean, I can think about all the ways that oxalate impacts host health, while sitting in my office, but it's really, it requires patient input in order to really understand what oxalate is doing for health. So I see this as a long-term relationship, both designing clinical studies and having people participate in it, and also having this long-term dialogue that we can really facilitate the design of future clinical studies and trials. And ultimately, we want to develop some novel therapeutics to help mitigate the impacts of oxalate for health. So just a little bit of background about myself. So I'm assistant staff at the Cleveland Clinic. I've been there since 2016. So my research career, so the first 5 years of my research really started in a completely different field, looking at anthropogenic associated causes of coral disease. But it was really during that period that I started to really appreciate biological organisms as complex systems. And because of that appreciation, that's really why I started my research into oxalate. And so the unique features of oxalate microbe-host interactions, which I've been studying now for the last 11 years, really allows us to interrogate some of the fundamental features of complex biological structures. And so that's what I'm really academically interested in. And then importantly, this research obviously has relevance for human health and disease. And so, like Susan said, I take a multi-omics approach to research that really spans animal, clinical, and in vitro systems. And so if you're unfamiliar with omics technologies, these technologies are really designed to profile all of something in a sample. So metagenomics looks at all of the genes or DNA in a sample. Metatranscriptomics looks at all of the gene transcripts. We have metabolomics that looks at metabolites, proteomics that looks at proteins, culturomics that looks at cultural bacteria, and the list really goes on. And so as you can imagine, these multi-omic technologies produce an enormous amount of data. And so we use a lot of bioinformatics to interrogate those high volumes of data. And really bioinformatic tools involve a lot of novel computer algorithms and programming that use cutting edge statistical approaches that can handle sort of the hyper variable data sets that are generated by that multi-omic technology. So my primary research focus is on oxalate homeostasis, but really because I am studying oxalate, this is expanded out to look at the urinary tract microbiome, on neurologic health, which in part is also focused on oxalate metabolism. And we also have expanded out to looking more at gut microbiota and dyslipidemia, which is also oxalate related, and I'll talk about that a little bit today. So this is kind of the star of the story and really where this research started. And so this is the white-throated wood rat called Neotoma albigula. And it's really unique in that it is a mammalian dietary specialist. Specifically, it consumes exclusively a punja cactus. And so this cactus is very high in oxalate content, but there's no known apparent negative effects of oxalate or this high oxalate consumption on these animals. And past research have shown that mammals do not harbor enzymes that are capable of breaking down oxalate. And so it really suggests that these animals harbor a unique gut microbiota in order to handle these high oxalate diets. So to start off with these animals, what we wanted to do is sort of determine what their threshold of oxalate tolerance was. And so basically what we did is we went out to the desert, like Susan said, and we caught these animals and brought them back into the lab, and then conducted some controlled dietary experiments on them. And so specifically, what we did is we put them onto a diet and gradually increased the oxalate content of that diet up to 12% of the mass of the diet. So 12% of the mass of the diet was oxalate, which, as you can imagine, is an enormous amount. And so what we did initially is look to see how much of the oxalate was being degraded by these animals on those different diets. And so what you can appreciate here, we have the amount of oxalate consumed by the amount of oxalate degraded. And what you can appreciate is we never reached that threshold of tolerance in these animals. Even at 12% dietary oxalate, these animals were degrading nearly 100% of everything that they consumed. And so that really points towards a really remarkable gut microbiota in terms of oxalate metabolism. So next, what we wanted to do is look to see if oxalate had an impact on the gut microbiota of these animals themselves. And so we employed this same dietary strategy, gradually increasing the amount of oxalate. And then we did some next generation sequencing or high throughput sequencing of the 16S rRNA gene. And so this gene is universally distributed amongst all bacteria, and it's commonly used to taxonomically classify them. And so it's used to profile the gut microbiota in multiple different study systems. And so what I'm showing here is the impact of oxalate on the number of species that we detected in these in this gut microbiota. And so what you can see here is as you increase, and hopefully everybody can see my cursor here, but as you increase dietary oxalate on the x-axis here, we see a significant increase in the number of species that we can detect through high throughput sequencing. And these letters at top represent the statistical groupings, showing that this is a statistically significant effect. And what this really suggests is that oxalate may actually benefit this gut microbiota by allowing some of the rarer taxa to proliferate and become detectable through our sequencing approach. Next, what we wanted to do is do the inverse and see if the gut microbiota impacted oxalate degradation or oxalate homeostasis. And so what we did is we developed a fecal transplant model, taking the stool of our wood rats and transplanting it into Sprague Daly rats. And as a note, all laboratory rodents are notoriously devoid of oxalate degrading bacteria. And so it makes an excellent negative control for these kinds of studies. So prior to our fecal microbial transplants, all of our Sprague Daly rats had a similar level of urinary oxalate excretion, shown here on the first column. But immediately after our fecal transplant, we saw a significant reduction in urinary oxalate compared to our negative control. And we also included in the study two probiotic formulations. One was a commercial probiotic called VSL number 3, which has some known oxalate degrading bacteria in it. And we also have oxalate degrading bacteria that we specifically also isolated from our Neotoma albigula wood rats. And while we saw a similar reduction between these three different treatments, the greatest impact was in the fecal microbial transplant rather than the oxalate degrading bacteria alone. This was followed by a no oxalate washout period, and then we put them back onto the oxalate diet to see if this function remained. And lo and behold, the one that functioned most effectively was fecal microbial transplant rather than the oxalate degrading isolates. And of course, our no transplant control had the highest level of urinary oxalate. So this tells us that not only does oxalate impact the gut microbiota, but the gut microbiota impacted oxalate levels. And so that's basically what we have here. So kind of to summarize those experiments, and importantly, we see that many species within the gut microbiota are responsible for oxalate homeostasis based on that measure of species richness. So next, what we wanted to do is to get a little bit more human relevance on this and interrogate the gut microbiota a little bit more. And so we want to look at the antibiotic and diet effects on oxalate homeostasis. So for this study, we had a similar fecal transplant model, though this time we switched to Swiss Webster mice, which are more genetically similar to wood rats, and it also allows for greater numbers and less time required for the diet studies. So in this particular study, we had 5 different treatment groups. So our negative control was a fecal transplant of Swiss Webster feces into Swiss Webster mice. And then we had either Swiss Webster mice with Neotoma albigula stool and no other intervention, or Neotoma albigula stool with antibiotics, specifically cefazoline, or a high-fat, high-sugar diet, or both antibiotics and a high-fat, high-sugar diet. And so these animals were maintained on a high oxalate diet for the period of the experiment. And we collected stool, both stool and urine over 3-day periods. And so what we saw is that immediately after the fecal microbial transplants, our Swiss Webster microbiota mice expectedly had very low levels of oxalate degradation. But prior to any additional intervention, the fecal microbial transplant animals had similar levels of oxalate degradation. But surprisingly, after only 3 days of antibiotics or the high-fat, high-sugar diet, the level of oxalate degradation dropped to our levels of the negative control compared to animals that had Neotoma albigula, fecal microbial transplant, and no additional intervention. If we look at urinary oxalate, we get similar results. So at right after the microbial transplant, animals receiving the Neotoma albigula stool had similar low levels of urinary oxalate compared to our negative control. But in this case, we had a more gradual increase in urinary oxalate for animals treated with antibiotics or the high-fat, high-sugar diet, compared to if we look just at oxalate degradation. And so what we can conclude from that study is that both antibiotic use and a high-fat, high-sugar diet reduced oxalate metabolism and increased urinary oxalate. And some of the data that I didn't show here is that this was specifically due to a reduction in the network of bacteria involved in oxalate homeostasis. And I'll talk a little bit more about those bacteria later. The study also reiterates the gut microbiota influence on oxalate homeostasis and has implications for antibiotic use and also diets for humans, for patients. All right. So next, we're going to get a little bit more into the systemic effects of oxalate. And specifically, this next study we designed in APOE knockout mice. And so these mice are typically used in studies of atherosclerosis. And so this is a more of a systemic vascular impact mouse study. And so for this study, what we did is we had APOE mice with their native microbiota, so no microbial transplants, on a basal 0% oxalate diet or a high 3% oxalate diet. And they had either no antibiotics or antibiotics in order to look at the effect of the gut microbiota. And so for the study, we looked at the gut microbiota. We looked at the lipid profiles of the kidneys, inflammatory cytokines, plasma oxalate, and urea. And we also looked at the aortas for atherosclerotic plaques, the kidneys for renal calcification and injury, as well as renal lipidomics. And we also quantified primary and secondary bile acid synthesis, which I'll explain why in a moment. So right to the heart of things, on these histopathological images, these show the aortic root of the hearts after an 8-week study on the different diet and antibiotic trials. On the top, we have our control animals, so a basal diet, no antibiotics. And this arrow here is pointing to the sliver of red. So this is the atherosclerotic plaque. And so you can see it's very, very tiny in these animals compared to the image on the bottom, which is oxalate and antibiotics. And so here you see a much larger red area, so a much larger atherosclerotic plaque in these animals. If we quantify the total area of the atherosclerotic plaque from these animals, we see that there's a significant oxalate and antibiotic effect. So both, so there were independent diet and gut microbiota effects on atherosclerosis. And so when I have these numbers up here, these represent the p-values of our different treatment groups. And so, of course, as you know, any values below 0.05 are considered significant. So next, what we wanted to do is try to understand a little bit more about why oxalate was promoting atherosclerosis. So on the left, again, we're showing the aortic root, and the red area is the atherosclerotic plaque. And using polarized light, we can visualize directly the calcium oxalate crystals that are in the tissue. And so that's illuminated here by these bright white crystals, whereas the rest of the tissue in the background is just black. And if we overlay these two images, what you can appreciate is that the atherosclerotic plaque has an enormous amount of calcium oxalate integrated into it, and calcium oxalate isn't present in the remaining of the tissue. And so again, if we quantify the calcium oxalate in these plaques, we see again that there's a significant effect of both dietary oxalate and antibiotics on the integration of calcium oxalate into the atherosclerotic plaques, which gives one potential mechanism about why oxalate promotes atherosclerosis in these mice. So in these same animals, we also looked at the kidneys because of the known effects of oxalate on kidney pathology. And so interestingly, what we found were two different oxalate-related phenotypes in the kidneys. And so animals that were given an oxalate diet without antibiotics, they had more of a kidney stone phenotype. And so this was indicative by, in this kidney here, we have a clear obstructive uropathy. And so when the kidney looks like this, I have a kind of an image down here where you have some obstruction in the ureter, and that creates this hydronephrosis that causes this expansion of the kidney. But if we had oxalate and antibiotics, we get more of an oxalate nephropathy or more of an oxalosis phenotype. And so that's really illustrated by this polarized light image of the kidney. So all of these white crystals here is calcium oxalate. And so you can see it's throughout the entire kidney tissue. And so we looked at multiple different metrics of kidney health as well as oxalate-specific kidney metrics. And so consistently, we see both an oxalate and a gut microbiota dependent effect of oxalate on kidney health. And so we also use these kidneys with transmission electron microscopy. And so specifically what we were interested in is the potential development of Randall's plaques. And so Randall's plaques are calcifications that occur in the subepithelial space of the loop of Henle and the nephrons of kidneys. And so it's thought that Randall's plaques are the progenitors for most human kidney stones. And so we wanted to see if the calcification we're seeing in these animals was similar to human-like kidney stone formation. And so on the top here, we have the lumen of a renal tubule. And I'm pointing out that the basal membrane, so this is the loop of Henle here. And on the bottom, we have a close-up of some calcifications that are in the subepithelial space of the basal membrane, showing here. Showing that the kidney stone-like phenotype that we're seeing starts with Randall's plaque in that subepithelial space of the basement membrane. So we wanted to get a little bit more insight into the oxalate interaction with kidney stones and the kidney pathologies. And so we did some lipidomics on the kidney tissue. So lipidomics is profiling all of the lipids using a mass spectrometry method. And so on the left here, so this is a peak-OA plot. And so just to kind of orient you, each of these points here represents an individual sample. And the distance between the points represents the dissimilarity in the presence and absence of lipids, as well as the relative abundance. And so based on the different groups of our study, we can do a statistical analysis on the whole lipidome. And so what we saw here, again, was a gut microbiota dependent and independent effect of oxalate on the kidney lipidome. And so you can see if we gave the animals oxalate without antibiotics, we saw a shift from our controls. And then this was greatly exacerbated when we gave these animals antibiotics as well, and shown in that blue circle here. In particular, there were three lipids that were significantly enriched in our oxalate group. And so specifically the ceramide, diacylglycerol, and triglyceride. And these are all strongly implicated in atherosclerosis and cardiovascular disease. So another important part of cardiovascular health are bile acids. So bile acids come into two different flavors. So primary bile acids are produced by the liver by host metabolic mechanisms. And then a number of these primary bile acids make their way into the gut, where they're transformed by gut bacteria into secondary bile acids. And so we performed an analysis of many different primary and secondary bile acids, and I'm showing just a few of them here. But we very broadly saw gut microbiota dependent oxalate effects on both primary and secondary bile acids. And so what this tells us is that oxalate is potentially having an impact on liver activity because of its negative effect on primary bile acids. But it also appears to have an effect on the gut bacteria because of the negative effect we see in secondary bile acids. And interestingly, particularly with I showed two of the more interesting secondary bile acids, the lithocolic and dioxycholic acid, because initially the oxalate alone actually significantly increased these bile acids, but both bile acids were completely wiped out with antibiotics. And so there are some differential impacts being shown here with oxalate and antibiotic use. So another important marker of atherosclerosis and cardiovascular health obviously is cholesterol. And so we also looked at plasma cholesterol and triglycerides in these animals. And so on the left, we're showing the total plasma cholesterol in these animals. And what was really interesting to us is that oxalate by itself did not actually impact plasma cholesterol. But when you gave these animals oxalate and antibiotics, we saw a very stark increase in cholesterol levels. And so there seems to be some interesting interactions between oxalate and cholesterol, and potentially some of the metabolites that are being produced due to oxalate exposure on the gut microbiota that influences cholesterolemia. In contrast, if we looked at the triglycerides, we didn't see any significant impacts. So it seems to be targeted towards the cholesterol specifically. We also did a glucose tolerance test on these animals, which is an important indicator of cardiac health and atherosclerosis. So if we go by the actual curve of the glucose tolerance test, one of the interesting things that we saw was the peak glucose levels in animals that were fed oxalate and given antibiotics was shifted compared to our other treatment groups. And if we look at, if we do a statistical analysis on when this peak occurs, we saw that there were significant differences with oxalate and antibiotics compared to the other groups. So it also impacted this glucose tolerance in these animals. So what we can summarize from this experiment is that oxalate exposure integrates into and increases the size of atherosclerotic plaques. And it also influenced renal dyslipidemia and renal pathologies, which were dependent, both of these were dependent on antibiotic use. So interestingly, oxalate impacted both primary and secondary bile acid production, which again were driven in part by antibiotic use. And all of these factors have important implications for both cardiac and renal health. So now, given what we saw in the gut microbiota and the liver with our previous APOE study, we wanted to dig a little bit more into the oxalate and the gut-liver axis. And so we followed a our similar fecal microbial transplant model that I talked about previously with our antibiotic and high-fat, high-sugar diets. And so again, we used Swiss Webster mice as our host, and then we had either a Swiss Webster microbiota or a Neotoma albigula microbiota. And then we had them on either a 0% basal oxalate diet or a high 1.5% oxalate diet. And at the end of the study, we performed necropsies and collected liver tissue to look at the total RNA using RNA-seq to evaluate hepatic gene expression. So in this PCOA plot, what the points indicate are the gene expression of the liver. And so the distance between them represents the dissimilarity in the presence and relative abundance of genes detected from our liver samples here. And so importantly, what we saw was that there was a constitutive microbiome impact on the hepatic activity. And there was a not quite significant impact of oxalate, but the reason for that is because there was an interaction between the microbiome and the diet. And specifically, if you look at the black and the blue circles, that there's a considerable amount of overlap. These are the animals with the Neotoma albigula microbiota, and then the orange and the purple are the ones with the Swiss Webster mouse microbiota. And so to kind of look at this a little bit further, we did a differential abundance analysis on the hepatic genes. And so what is abundantly clear here is that in animals that had the Neotoma albigula microbiota, there is very little change in hepatic activity with exposure to oxalate. However, in the animals that had the Swiss Webster microbiota and exposed to oxalate, we saw a considerable increase in the number of genes being expressed in the liver. And so this suggests that in these animals, when you expose them to oxalate, their liver activity increases pretty considerably. And so we looked at the specific genes that were altered by oxalate in these two different mouse models. And what it really boiled down to was that oxalate was having an impact on the mitochondrial form and function in our Swiss Webster mouse model. And so this corroborates past studies that shows that oxalate primarily targets the mitochondria in liver cells, and shows that the increase in liver activity is primarily relegated to the mitochondria. So next, what we wanted to do is look a little bit more specifically at the oxalate effects on microbial metabolic output. And so we used that same animals that I just showed you in the last slide, but in this case, we took an untargeted metabolomic approach to look at the metabolites present in the guts of these animals. So again, what we see is that there's a very strong constitutive impact of the gut microbiota on the metabolites that are present, perhaps unexpectedly. And so in the top left, we have Neotoma albigula, and the bottom right, we have the Swiss Webster microbiota. And then similar to our liver gene expression, we didn't see much of a difference in the metabolites with oxalate exposure in animals that had the Neotoma albigula microbiota. But we did see much more separation in animals that harbored the Swiss Webster microbiota. And so we did a similar differential abundance analysis. And so while the differences between our hosts with the Neotoma albigula microbiota and Swiss Webster microbiota are not as stark as what we saw with liver gene transcription, similarly, we see much less of an effect of oxalate on changing the microbial metabolic output when animals had the Neotoma albigula microbiota compared to those that had the Swiss Webster microbiota. But importantly, we actually saw a net increase in metabolites with the Neotoma albigula microbiota and a strong net decrease in metabolites with Swiss Webster mice. So this again suggests that with the Neotoma albigula microbiota, that oxalate may be beneficial, whereas in our Swiss Webster microbiota, oxalate seems to be inhibitory towards microbial metabolic output. So if we look at the specific metabolites that are altered by oxalate in these two different mouse models, primarily comes down to secondary metabolites. And so secondary metabolites produced by bacteria are not essential for growth and reproduction, but they're really geared towards adapting to environmental changes. And so they provide a competitive advantage in a dynamic environment. And so it really shows that what's being hindered here in our Swiss Webster mice is this adaptability by the bacteria. So these two transcriptomic and metabolomic studies were done in the same mice. And so then what we did is to integrate this data in order to directly look at how microbial output with oxalate exposure impacted liver activity. And so this first network, so this is looking at the significantly differentiated microbial metabolites correlated to the significantly differentiated liver gene or expressed genes. And so in animals that had the Neotoma albigula microbiota, there wasn't a big change. And so we had 58 metabolites that were different, or 22 hepatic genes or that were correlated. And by and large, this was a negative interaction. So this means that the microbial metabolites that were produced due to oxalate exposure led to a decrease in liver activity overall. This is in stark contrast to animals that had the Swiss Webster microbiota. So we had far more interactions between liver genes and microbial metabolites. And importantly, there were 3 times the number of positive interactions than negative. And so what this means is that by and large, the metabolites that were produced with oxalate exposure by the gut microbiota led to an increase in liver activity. And so what we can conclude from this is that oxalate induces a strong hepatic response in the host, targeting specifically the mitochondria. Oxalate inhibits the metabolic output of the gut microbiota for those that don't have oxalate-degrading bacteria. However, with a strong oxalate-degrading community, this eliminates the negative effects of oxalate on both the liver and the gut microbiota through host-independent mechanisms. And then oxalate stimulates diverse Neotoma albigula microbes, but it did not significantly alter metabolic output. And so what this really tells us is that there's a lot of metabolic redundancy amongst diverse microbial taxon. And so this is something that we wanted to explore further next. So next what we want to do is look at a little bit more specifically of how oxalate impacts the gut microbiota. And so we went back into our Neotoma albigula model, so with a Neotoma albigula host and microbiota. So this was a shotgun metagenomic analysis. And so shotgun metagenomics looks at all of the genes that are present within that sample. And so not surprisingly, similar to our taxonomic profiles, we saw a significant difference in the microbial genes that were present with oxalate exposure. So again, showing us that oxalate stimulates diverse bacteria within the Neotoma albigula microbiota, but again, without changing metabolic output. And so we wanted to look specifically what these genes were. And so I have them all listed here, but what it really boiled down to were specific pathways. And so it's important to point out that the byproducts of oxalate degradation are primarily formate and CO2. And so these substrates can be used in additional microbial pathways, and specifically methanogenesis, acetogenesis, and sulfate reduction. And so when we look at the pathways of these microbial genes, many of them mapped to the methanogenic, acetogenic, and sulfate reducing pathways, in addition, of course, to oxalate degradation. But there also seem to be a lot of sort of commensal pathways and like sugar metabolism genes that were also stimulated by oxalate. And so to kind of target the taxa that were associated with this response a little bit more finely than just our 16S taxonomic surveys, we took that shotgun metagenomic data and we extracted bioinformatically full-length genomes. And so what I'm showing here is a phylogenetic tree of all of the genomes that we extracted. And so the entire phylogenetic tree represents the bacterial tree of life, and the blue stars represents our genomes. And so what you can appreciate here is we have a very broad diversity of genomes that we extracted from these animals. And so first, what we wanted to do is look at a little bit more specifically what taxa were responding to dietary oxalate. And we did this by mapping the significantly different genes back to our full-length genomes. And so unsurprisingly, 3 out of the 4 taxa that were most stimulated by oxalate were different strains of Oxalobacter formigenes. So these strains are known oxalate specialists. They require oxalate as a sole carbon energy source, but we also had many from Allostipes and Mirabaculi, which are abundant gut bacteria that are also known oxalate degraders. We also use these genes to quantify oxalate metabolic redundancy. And so we found a large proportion of the genomes harbored at least one oxalate degrading gene. And these were primarily distributed amongst oxalo-CoA and formal-CoA genes, which are the most commonly focused on oxalate degrading genes in the gut microbiota. But there were diverse other oxalate degrading genes that were present in the gut microbiota here. Importantly, these genes were distributed across many diverse taxa within the gut. And specifically, we found that nearly 60% of all the genomes that we looked at had at least one oxalate degrading gene. We also repeated this same analysis in a human population, and we found that 35% of genomes extracted from the human gut microbiota contained at least one oxalate degrading gene. And so this is a lot lower than that 60%, but still far greater than what we had originally anticipated. And what it really highlights is that oxalate metabolism seems to be a very critical function for bacteria in the gut for survival. So up until now, we really focused on whole gut microbiota analyses for how oxalate interacts with the microbiome and the host. And so next, what we wanted to do is try to refine a specific small microbial community that can maintain oxalate homeostasis. And so we've conducted a number of clinical and animal studies to really try to identify what species are important for oxalate homeostasis. And so through those studies, we came to a small consortium of 8 specific species that we thought were really important for oxalate metabolism. And so we brought these bacteria into the lab, and we defined 4 different groups. And so as a control, we used our whole Neotoma albigula community. And then this ox group is Oxalobacter formigenes by itself. And then we have Ox4, which includes Oxalobacter and acetogenic, methanogenic, and sulfate-reducing bacteria. The all group also contains some commensal bacteria. And then no ox is all of our bacteria minus Oxalobacter formigenes. So we want to remove that known oxalate degrader. And what you can appreciate here is, so this is an in vitro analysis of just bacteria and oxalate metabolism. And so if we include Oxalobacter, which is a known specialist, we see oxalate degradation values that are through the roof, this is expected. But even if we remove Oxalobacter formigenes, we saw significant levels of oxalate metabolism that were on par with our positive control, the whole community from Neotoma albigula. So this, again, really suggests that there's a lot of metabolic redundancy within these bacteria that commonly come up in our animal and our clinical surveys. So because of this response, we wanted to tease apart which specific bacteria were responsible for the oxalate degradation response. And so I have here listed the different bacteria that are included in this all cohort right here. And surprisingly, all but one of our oxalate, or all but one of our species that we included in this consortium degraded significant amounts of oxalate. But importantly, this was highly dependent on whether or not formate was present within the media. And we also looked at formate metabolism within these species and found that multiple species also utilize significant amounts of formate. But again, this was highly dependent on the presence or absence of oxalate or some of the rich components of the media that we used. So we wanted to look a little bit more at the metabolic cooperation hypothesis. So we have an abundant amount of evidence that shows that oxalate metabolism is highly redundant to surprising levels. But we wanted to target this metabolic cooperation a little bit more. And so we repeated this oxalate degrading analysis in vitro with our different groups of bacteria. And again, what we found is that when oxalate degrading, or when Oxalobacter formigenes is by itself, it degrades a pretty considerable amount of oxalate, as does our other combinations of bacteria. But next, what we wanted to do is we wanted to look at media that contained only oxalate as a sole carbon and energy source, and then quantify the amount of formate in the media. And so, as expected, when Oxalobacter was by itself, it produced an enormous amount of formate as a result of oxalate metabolism. But when we include our acetogenic, methanogenic, and sulfate-reducing bacteria, that formate level that's produced specifically by Oxalobacter was reduced pretty considerably. If we have all of our bacteria, including our commensals, that was even less than with our just the acetogenic, methanogenic, and sulfate-reducing bacteria. And then even when we remove Oxalobacter, there's a little bit of formate that's produced, and that's also eliminated. And so then this, again, is strong evidence that not only is there metabolic redundancy with oxalate degradation, but we also have clear evidence of cooperation, specifically in the formate that's produced by oxalate degradation gets utilized in these other metabolic pathways. So next, what we wanted to do is bring these bacteria into an animal model. And so we used our Swiss Webster mice with our microbial transplant system. And so similar to our fecal transplants, what we saw is that when animals were not given a microbial transplant, we had very high levels of urinary oxalate. But in all 4 of our microbial transplant communities, we saw a significant reduction in urinary oxalate. And this was, when we looked at urinary formate, we saw something similar to our in vitro results. And specifically, when Oxalobacter was there by itself, the amount of formate that was produced was much higher than our other microbial transplant communities. And then only when we include both Oxalobacter and our acetogenic, methanogenic, and sulfate-reducing bacteria do we see this stark decrease in urinary formate levels. And we also looked at the effect of these microbial communities on renal calcium oxalate deposition, which are indicated by these black areas here. And all 4 of our microbial community transplants had significantly lower levels of renal calcium oxalate deposition compared to our no microbial transplant negative control, showing that all of these bacteria in some way contribute to reducing calcium oxalate deposition in the kidneys. So then what we wanted to do is look more at the intestines. And so obviously, oxalate is introduced into the system through the colon. And so we wanted to get a better idea of how oxalate is impacting the colon itself. And so this is a graph of fecal oxalate levels. And so similar to our urinary oxalate, the animals receiving all of our microbial transplants had significantly lower levels of fecal oxalate compared to our no microbial transplant control, with the greatest effect being in animals that had all of our bacteria or Oxalobacter with our acetogenic, methanogenic, and sulfate-reducing bacteria. So here we have some histopathological images of the colon tissue. On the top, we have our no bacteria negative control showing significant amounts of colitis. And particularly this kind of area in the top kind of center, these are inflammatory cells, inflammatory infiltrates into the tissue. Whereas the bacteria that had all of our, or sorry, the animals that had all of our bacteria, we had much more of a normal looking colon. When we could quantify the colitis phenotype, again, all 4 of our microbial transplants had a significant effect on reducing colitis. And so these data are important in showing both that oxalate directly impacted colitis and that our bacteria can alleviate that effect in these animals. Then we looked further into the heart of these animals, kind of building on our atherosclerosis study. And so specifically in this stain, we're looking for cardiac fibrosis. And so the dark areas here are the fibrotic areas of the heart tissue. And so you see our negative control on the top showing a lot of these dark blue areas versus the animals that had all of our bacteria showing virtually none. Again, when we quantify this, all 4 of our microbial transplant groups was able to significantly reduce the oxalate-induced cardiac fibrosis. So to summarize this bit, we saw through a series of animal studies that by far and away was most important for oxalate homeostasis was oxalate metabolic redundancy. And specifically, this improved renal calcium oxalate deposition, colitis, and cardiac fibrosis. But we also saw some significant oxalate-formate metabolic cooperation, specifically where formate produced by oxalate metabolism provided some carbon and energy sources for other acetogenic, methanogenic, and sulfate-reducing bacteria, specifically when other resources were already used up. And so if these bacteria can use some of the rich compounds in the environment, then they preferentially use those. But if formate's the only thing available, then that's what they'll use. So to kind of wrap up the talk, we see that oxalate impacted multiple organs. This included the heart and the vascular system, the liver, the colon, the kidneys, and the gut microbiota itself, which many people consider it its own organ. And we also see that microbial metabolism was driven primarily by metabolic redundancy, but we also see clear impacts of metabolic cooperation within the gut. So like I said, this is primarily in animal studies. And so we do a lot of clinical studies as well. And so we're actually wrapping up a clinical intervention study in participants that were not colonized by Oxalobacter fumigines. And then when these participants were given Oxalobacter as a probiotic, similar to many clinical trials in the past that have used Oxalobacter, only a small proportion of our participants actually responded to Oxalobacter by lowering urinary oxalate. And when we looked into the gut microbiota of these animals, what differentiated the responders and the non-responders was actually the presence or absence of other oxalate-degrading bacteria in the gut. And so this really coincides with a lot of the animal work that we're doing now and showing that the research into our animal models is directly relevant to what occurs in humans as well. So with that, I'll stop and I'll be happy to take questions. Thank you very much.
Okay, I will just suggest that if you have a question that you unmute yourself and then everybody else stay muted. And that way, and also there's a way that you can wave your hand. Down here, I'm trying to think about what reactions. You see the reactions? If you like click on the hand, then we'll see you.
Right, I think Chris.
Hi, thank you, Dr. Miller. I have 2 questions.
Yeah.
When you say that you gave the wood rats oxalate up to 12% of their diet, is there a way that you can equate that to what a human would be eating in milligrams of oxalates?
Yeah, so that level of oxalate, if a human were to consume the same number of milligrams, that would be considered like a high oxalate diet, so around like 200, 250 milligrams. And keep in mind, like these are tiny animals, so like to them it's very, very high. But so it would be considered a high oxalate diet for humans.
Okay, so I'm going to repeat that. So 12% would be the equivalent of a human eating 250 milligrams of oxalate a day.
Yes, so it would be like a 12% oxalate diet would be similar to that.
Okay.
I just want to throw out that we did a survey, which was shocking to me, of the people that had autoimmune disease that were on our group. And we asked them what their diet, how high their diet was in oxalate before they found us. And there were a shocking amount of people who were over 1,200 milligrams a day. huge group.
Yeah, that's I mean that's a considerable amount. Like in the clinical study that I just briefly talked about, like we defined it was a controlled diet study in humans, and we defined high oxalate diet at like between 225 and 250. So like 1200 would be, yeah, it'd be a very high oxalate diet, I think.
I'll show you the results of that. Okay.
Yeah.
I'm gonna ask my second question, but first I'm gonna say that I just saw, like, I ate 2,000 to 3,000 milligrams a day because I was doing 3 cups of spinach. Like, this is very, very common. It's happening all the time. The moms, the Instagram people, the Pinterest people, they're all telling everybody to eat 2 to 3 cups of spinach 3 times a day to detox. So I'm just gonna drop that out there. Many of us who have lived it, they know. But my other question was, you showed that there was plaque in the heart, decreased bile, liver acids. This is all with antibiotics and oxalate. Increased cholesterol, and it targets the mitochondria. My question is, because I have a history of kidney issues, but the kidney people always say, and in the literature, it's what I read, of course, I don't read everything, right? That this type of accumulation only happens when there's a decrease in kidney function. So, and you didn't talk about it. And to me, this type of accumulation happens regardless of like kidney failure. Could you speak to that?
Yeah, so oxalate, I mean, it's kind of a chicken and egg question, right? So oxalate by itself does impact kidney function. So it can, if you have chronically high levels of oxalate going through your kidneys, then that by itself does reduce kidney function. But lower kidney function is not a prerequisite for accumulation of calcium oxalate in the kidneys. And so, I mean, we know this from people that have kidney stones, right? Like, I mean, not everybody that has kidney stone or calcium oxalate stones has low kidney function. I mean, like that's really kidney stones, like having recurrent kidney stones over a long period of time is what leads to reduced kidney function. And so, yeah, it's like lower kidney function is not a prerequisite for calcium oxalate accumulation.
I wanted to hear that. I wanted to hear you say that. Thank you.
Yeah.
Well, our doctors need to hear that.
Our doctors seriously need to hear that.
Yeah, and I mean, and certainly, like if you have lower kidney function, then that would lead to higher levels of accumulation because you would have the calcium oxalate sticking around longer, and you would have higher concentrations. But I mean, it's still kidney stones form with normal function kidneys.
So Ellie, I think we're maybe ready for you.
Thank you. Thank you so much, Dr. Miller, for coming to talk to us today. I really appreciate it. I've been trying to solve this oxalate problem with myself for a couple of decades now, and really happy for our Trying Low Oxalate group. Do you think, I think I have two questions. Do you think that we will be looking at fecal transplants as a solution for humans?
So no. So fecal transplants, they are a solution, but it's a very untargeted kind of like black box kind of solution. And so really what I'm trying to do, and I think a lot of people in microbiome research are trying to do, is have much more nuance. And so that's one of the reasons why we really tried to understand what specific species were involved in oxalate homeostasis. So I see more of a targeted microbial transplant as a means to mitigate oxalate homeostasis. And specifically, like what I alluded to, is we know from that last clinical trial and our the animal studies that I talked about, we know what would lead to somebody responding to Oxalobacter or another Oxalobacter therapy, right? And specifically these other oxalate degrading bacteria. And so if we can like provide a screen for that, then like we would be able to say like who can respond and who wouldn't. And so I think there's going to be a lot more nuance in those kinds of therapies.
Thank you. And then my second question is, as you develop this solution, how do you bring it to scale so that while I'm here in Marietta, Ohio, my doctor's like, oh yeah, Ellie, what you want to do is this transplant with this specific portfolio of microbes. I know just how to help you with that. Or will we need to have like the Center for Oxalate Healing at the Cleveland Clinic where we all go to the Center for Oxalate Healing?
Yeah, so what I see happening, so like there are two potential causes for, these oxalate effects, right? So one is the genetic and one is the microbiota. And so we're conducting a study right now to differentiate that in people that have very high levels of idiopathic hyperoxaluria. So what I see moving forward is people that have hyperoxaluria, we would do a simple screen of their gut microbiota to see if they have actually reduced levels of these oxalate degrading bacteria and see if they would benefit from our oxalate degrading community, or if it might be more of a genetic thing. And so we're actually moving towards testing, Oxalobacter and some of these other bacteria in clinical trials. But I see it as not like, something that can be done at home. Like we have from the clinical study that I mentioned at the beginning, we already have in place a validated at-home collection kit that you can, collect stool, collect urine, and send it back to us, within 24 hours, and we can, perform whatever screening or assays that we need to do. So that's kind of where I see it going, is having more of a standardized screening / like probiotic approach.
Thanks.
You're welcome.
Okay, Cynthia.
Hi, Dr. Miller. That was a great talk. Thanks. I just wanted to point out, I think it's so important that you were looking at atherosclerosis and colitis, because it's so generally accepted that oxalate affects kidneys. But Susan in the group has been trying to teach that oxalate can deposit all throughout the body in different organs and different tissues. So that was really interesting. And I wonder about oxalyl CoA needing thiamine as a cofactor. So we know antibiotics will hurt thiamine chemistry. And you were talking about when you gave Oxalobacter probiotics, there were responders and non-responders. And I just wonder how much you think something like the nutrients that we need as cofactors for oxalyl CoA matter to the microbiota, because we do have a lot of group members that say, hey, I did a microbiome test, I've got tons of Oxalobacter, but I still have oxalate issues. So I just think,
yeah, I mean, so that's a really good point, and something that I haven't looked at a lot specifically. So like I said, in some of the metabolomic data, we know that oxalate has a huge impact on the microbiota, and there are a lot of kind of gray areas that we don't have good insight on yet. So I pointed to several points of data where, there's an oxalate effect, and there's an antibiotic effect, but like they might be in opposite directions. And so we know that, from data like that, when you give antibiotics to the gut microbiota, the oxalate handling changes pretty dramatically. And so I'm sure there's a lot of other factors that are involved in this that we haven't really, touched on yet. And that's really kind of, where we're moving towards. We're doing a lot more of these kind of diet studies, these controlled diet studies in animals, and also, like I said, in humans as well, to hopefully kind of like get at those kinds of nuanced details.
Yeah, that's great.
One of the things that I would like to see, because, I don't know if Dr. Miller, this history about me, but I nearly died of bone marrow failure when I was 13 years old after I was given an antibiotic called chloramphenicol.
Yeah.
And definitely, I am very suspicious that it knocked out my thiamine chemistry in some way. But what we really need to have somebody do is to go through and look at each kind of antibiotic that's used in human beings and look at what chemistry does it stop, or which microbes does it kill that make nutritional things for us. And so I would just love to see that kind of study.
Yeah, and so we've done studies now, like, I mean, we look a lot about antibiotics, and we found associations with past antibiotic use and kidney stones, and like several other groups have too. But we've also found it with cardiovascular disease, with diabetes, with chronic kidney disease. So we know that antibiotics have a very strong effect on the microbiota and associated host health. And what I always tell, I mean, a lot of times when I go to research conferences, there's like a small handful of PhDs like me, and then like a bunch of physicians. And so what I always say to them is that antibiotics simply aren't a no consequence kind of therapy, right? And so I always say, like, yeah, if you're at risk of an acute infection that might be fatal, sure, give antibiotics. Like, I'd rather get a kidney stone than die. So, like, yes, prescribe antibiotics when they're absolutely necessary, but be smart about it, right? You don't need to give them for, like, if somebody has a viral infection, or if, like, there's many times that antibiotics are given for reasons that don't really matter, that they don't need antibiotics for. And so it's really, advocating for like smart antibiotic stewardship, and recognizing that it's not a no consequence kind of therapy, because a lot of physicians treat it as it is. And so that's what I always tell physicians when I give these kinds of talks.
Well, if we had data on the effects of different antibiotics on nutritional factors, those are things that we can supplement. So, I mean, that's kind of the easiest thing to do, is to figure out, oh, well, if you are hurting the microbes that make thiamine, then just give thiamine, that's pretty easy.
And so what are the, like what I talked about at the beginning, sort of the unique features of oxalate microbiome host system really lends itself to kind of tugging at these strings of the complex biological system. And so, like, what we're showing with these, the idea of this metabolic redundancy and metabolic cooperation, and how oxalate is really a critical function for the gut microbiota. These kinds of things can be directly applied to any number of dietary molecules, right? And so, kind of like to your point, we can do the same kinds of studies with thiamine or any other molecule, and we can say, oh, it's these bacteria that are responsible, like you need metabolic redundancy, or you need this feature or whatever. And so I think those, like what we're doing here, are easily translatable to a lot of other factors that maybe are directly or indirectly related to oxalate metabolism or other host effects, right? And so, like, we can do those kinds of experiments now.
Wonderful. You must have another question, Cynthia. No?
No, that's everything.
Okay, take away your hand, and then we'll see if anybody else has one. Okay, any other questions, anybody? Chris?
You see research moving to look at the impact of those of us who are consuming in the thousands of milligrams of oxalates?
So I think certainly that would be valuable research to do. So, like I said, right now we're doing a clinical study on people that have idiopathic, like very, very high levels of idiopathic hyperoxaluria. So, like more than 80 or 100. So, like the threshold for hyperoxaluria is like 40 milligrams per day. And so, like we're these are idiopathic sources, so we don't know if it's diet or microbiota or genes, and we're kind of trying to figure out, like, in these cases, like what fraction is it. And I think looking at people that have very high oxalate consumption, relative to their urinary oxalate levels or their systemic oxalate levels and their gut microbiota, I think would give us some valuable insight too, and at least help us to devise therapies that might, if there's an issue, devise therapies that can help to mitigate those, even with a high oxalate intake.
I was thinking that one mitigation would be to tell people to not eat over 250, but that's just me.
Yeah, I mean, certainly, I think that's true. And I mean, like I said, there's a very wide variability in people's gut microbiota and their ability to handle oxalate, right? And so, I mean, one of the issues too, so depending on whether high levels of systemic oxalate are microbial in nature or host genetics, if they're microbial, then if you have a moderate amount of oxalate, then I see that as supporting those oxalate degrading bacteria. So like we and others have done numerous studies where we increase oxalate and we see an increase in oxalobacter and some of these other oxalate degrading bacteria. And so like people that have very high oxalate loads, yeah, I mean, I would absolutely, say like maybe reduce that to a large degree. But I would also I see value in some oxalate, right? Because that supports some of those oxalate degrading people or oxalate degrading bacteria too. And I think, a lot of, what we see in the group here, the TLO group, kind of, echoes that to some degree too. And echoing the idea that the oxalate reduction needs to be modest and at least very slow, right? So yeah, it's a very nuanced kind of question.
Thank you.
Thanks.
Any other questions? Let's see that we had one that came through the chat. Let's see if I can get that. Do you know why some people will develop nodules in thyroids, most likely from calcium and oxalates?
So I mean, it depends. So like in the literature, they really point towards oxalosis as kind of like where you get calcium oxalate kind of everywhere, right? But in our studies and what we're starting to appreciate more, and like Susan, what you have said many times is that oxalate can accumulate everywhere, even at even when you don't have oxalosis. And like I mean, there's studies on breast cancer tumors, and there's actually a couple of studies in calcium oxalate and atherosclerosis now. So I can't, I guess, speak to that specifically, but we do know from the studies that I talked about today that oxalate generally induces inflammation kind of throughout the body. We see that in multiple organs, and so I would suspect that it's related to that. And we do know that it accumulates in tissues, and we're looking a lot more at multiple different tissues in our studies now, trying to see that oxalate integration in other areas as well. So hopefully we'll be able to answer those kinds of questions soon too.
Right. Well, one of the things I wanted to say is that, we want to be as helpful as we can be to your efforts, because you're definitely studying the things that we want you to. I mean, it's like you're Santa Claus to us because you're doing exactly the kind of work that we know absolutely needs to be done. And so just whatever way we can be useful to your group, we volunteer ourselves to be useful. If you need to use us to put together a study of people that are reducing oxalate, or just whatever is in your mind.
Yeah, I mean, like I said at the beginning, I see this as kind of a a long-term relationship, and so we've worked for a number of years now to get these at-home collection kits validated and approved for IRB, and we have IRB approval now for social media recruitment and stuff like that, and so we have like all the pieces in place, and so I see this as kind of a long-term thing where we can really look at some of these impacts of oxalate. And obviously, like a group like this, they're highly motivated, and they know what oxalate is and they're interested in it. So yeah, I mean, I definitely see some interaction, in the future and with multiple studies, not only in ones that we have ongoing, but also in kind of figuring out how to plan future studies and what's really important for patient outcomes and things like that. So I definitely see this as kind of a long-term working group.
We know it's shocking, really, that some of the major health issues that people worry about, like dying of a heart attack or having atherosclerosis and having a stroke, or all of these, we dread it at the end of our lives that it gets worse and worse and worse and worse. Obviously, oxalate's something that if it accumulates in the body, it's going to be worse as you get older, right?
Yeah.
And so what you showed us today, Dr. Miller, is just incredible when you think about the implications to health and to other ways of, we've kind of let pharma say that, we're going to fix atherosclerosis in this way or that way. They have different drug solutions for it. But if you're not really getting to the cause, then you're not going to really solve the problem. And so we appreciate so much how you are getting to the reasons why these things are happening to us.
Yeah, thanks. And we also have a clinical study now for atherosclerosis too, and looking at oxalate and other dicarboxylic acids, because I mean we really think that oxalate plays a strong role in why cardiovascular disease and obesity and diabetes are comorbid with kidney stones and chronic kidney disease. We think it's all related to the gut microbiota and to oxalate and similar sorts of pathways, and that's what we're really trying to get at.
Well, it's kind of a Venn diagram, and you have the overlap that some people have these issues and some people have those, and then there are a lot of people that are in the overlap zone.
Yeah.
But there are people that maybe it's only going to manifest in one way rather than a different way. And so what our group I think is most useful in looking at is the people that don't have kidney stones.
Right, for sure.
And that would be so wonderful to see that firmly said out there in the academic world, that there are people that have oxalate issues but don't have kidney stones. And our study that we did on autism, it was done by some people that were used to handling people with primary hyperoxaluria. So they were that sort of research team. And we looked at the risks of getting kidney stones in autism, and it was basically zero, even though they had very high levels of oxalate.
Yeah, I agree. And one of the things that we're doing in our kidney stone research, kind of looking beyond oxalate, we've done a number of metabolomic screenings of urine and kidney stones themselves, and we've done a lot of microbiome analyses. And there are strong contributions of specific metabolites that outside of oxalate and specific bacteria. And so, like to me, based on our clinical data, what manifests as a calcium oxalate kidney stone requires not only high calcium oxalate, but also specific bacteria and also specific metabolites that we think are also produced by these bacteria. And so if you don't have those bacteria, you're not likely to get kidney stones. It might, like the oxalate might manifest somewhere else, like in atherosclerosis or some other phenotype, right? So there are other contributing factors that dictates whether you get phenotype A or phenotype B or phenotype C, right? But it's not all just kidney stones.
Well, our group is exploring all these things because really what we look at is, as we reduce oxalate and we get better, what changed? And so obviously the things that changed are the things that you think, well, maybe the oxalate was pushing that in the first place.
Yeah.
Make sense?
Yeah, no, I completely agree. And it's certainly like there needs to be a lot more research in this space. And obviously like it's slowly starting to branch out of, outside of urology and stuff like that. There are, a handful of, pretty decent, robust studies that shows oxalate effects elsewhere. And that's certainly like where it's moving towards.
Well, we are so excited about your work, and we're so excited about the idea of a collaboration with our group and your researchers there at Cleveland Clinic. And I think the future looks bright.
For sure. Thanks.
So thank you so much for coming to us today. And do you have any final words?
Not really. I just wanted to thank everybody again for joining, and thanks for the invitation to talk, and look forward to future interactions.
Okay, well, thank you. Well, you have a good rest of the day, and we'll tell our European people have a good evening, and we'll tell our Australian people to have a good day. Okay, good night, everybody.
Yeah.